A saliva sample from an Anishinaabe woman arrives at a laboratory. Her grandmother survived one of 526 federal boarding schools in America. Trauma lives in her body, passed down through generations unspoken. But the sequencer finds her, written in her granddaughter's DNA. The past is not past. It is biological. This is a story about Indian boarding schools and what they left inside living human bodies. In 2025, the National Native American Boarding School Healing Coalition confirmed 526 schools. That is more than one school for every single day of the calendar year, spread across 37 states. If you drove to one per day starting January 1st, you would not finish until late June. But this video is not primarily about the schools as buildings or policies. It is about what those buildings did to human biology and kept doing long after they closed. The mechanism is called epigenetic transmission, and the data emerging from peer-reviewed research is further reaching than historians expected to find. We're going to walk through that data carefully, gene by gene, cohort by cohort. The school's curriculum directly altered children's neurological development and stress responses. Not metaphorically, physiologically. When a 7-year-old Anishinaabe girl was separated from her family and placed in an institution, her body registered an emergency. The hypothalamic pituitary adrenal axis, the brain's central stress command system, activated. Cortisol flooded her bloodstream. Cortisol is the body's primary stress hormone, the chemical alarm that prepares you to fight, flee, or freeze. A single cortisol surge is normal, healthy even. That is the system working correctly. But when that alarm runs continuously, for months, for years, something else begins to happen. The body starts editing its own instruction manual. That editing process is what scientists call epigenetic modification. Here is the mechanism researchers documented in boarding school descendant studies. There is a gene called FKBP5. FKBP5 regulates the body's glucocorticoid response, essentially, how sensitive you are to cortisol. Think of it as the volume knob on your stress response system. Under chronic, inescapable stress, the body chemically tags that gene through a process called methylation. Methylation is not a mutation. It does not change the underlying DNA sequence. Instead, it attaches a small chemical marker, like a sticky note on a page, that tells the cell how to read that gene. Under chronic stress, that volume knob on cortisol sensitivity gets permanently turned up. The stress system recalibrates for a world that is always dangerous, always threatening, never safe. Now here is where the science becomes significant and difficult to sit with. Those methylation tags are heritable. They can be passed from parent to child through the sperm and egg cells before the child is ever born. The child does not need to experience the original trauma. The child does not need to know the original trauma happened. The body already knows. The gene has already been tagged. Inherited. Without consent. Without memory. This is the thermostat model that researchers use to explain epigenetic trauma transmission. Every human being is born with a biological thermostat that regulates stress response. Normally, that thermostat gets calibrated in early childhood by the environment you are born into. But the boarding school system reset the thermostats of entire generations under extreme, institutionalized duress. And those recalibrated thermostats were then installed, biologically, in every child born afterward, whether those children ever set foot near a boarding school or not. The policy's biological consequence arrives as inheritance. The foundational cross-generational epigenetic model for this kind of research came from Holocaust survivor studies. In 2016, Dr. Rachel Yehuda at the Icahn School of Medicine at Mount Sinai published a landmark study. The study appeared in the journal Biological Psychiatry. Yehuda's team examined FKBP5 methylation in Holocaust survivors and their adult children. They found measurable methylation differences in the children, people who had never experienced the Holocaust directly. The biological signature of extreme, prolonged institutional trauma had crossed a generation. That finding established the cross-generational transmission model that Indigenous health researchers then applied to boarding school descendant cohorts. The question became, if this mechanism exists in Holocaust survivors, Does it exist here too? The answer, when researchers went looking, was yes, and it extended further than anyone had anticipated. In 2019, researchers at the University of Michigan School of Public Health published a foundational study. They examined Anishinaabe adults whose grandparents had attended federal boarding schools. Compared to community members with no boarding school lineage, these individuals showed elevated methylation of the FKBP5 gene. Their stress response systems were running differently at the molecular level, not because of anything they had personally experienced, but because of what their grandparents endured decades earlier. A saliva sample, collected decades after the last school closed, carries a chemical tag placed there by institutional policy. The laboratory equipment is reading history. Cold, precise, undeniable. Then, in 2022, a more comprehensive study went one generation further. Researchers examined 3 intergenerational cohorts of boarding school descendant families, grandchildren and great-grandchildren of survivors. Measurable alterations in cortisol regulation persisted across all 3 cohorts. The biological signal had not faded. It had traveled. If your great-grandmother attended a boarding school, researchers are now documenting that your stress biology may still carry the molecular signature of her experience. 3 generations of descendants, 526 schools, 150 years. That is not history. That is a present-tense public health event. But what the science found when it looked at how cortisol actually behaves in these descendants, that part changes everything. The specific measurement researchers use is called the cortisol awakening response. In a healthy stress regulation system, cortisol surges sharply in the first 30 minutes after waking. That morning surge is critical. It activates the immune system, sharpens cognition, prepares the body for the demands of the day. In descendants of boarding school survivors, researchers document a blunted awakening response. The morning surge does not arrive sharply. Instead, the baseline cortisol level is chronically elevated, meaning the body never fully comes down from alert. It is as if the system learned, before this person was born, that rest is not safe, that relaxing the alarm means vulnerability, that the threat is always present, always imminent. The body is defending against a crisis that ended decades before this person drew their first breath. Consider a Lakota child participating in one of these saliva cortisol collection studies. A great-granddaughter of a survivor from the era when Carlisle Industrial School was still operating. Her morning cortisol sample shows the blunted awakening response researchers have come to recognize. Her body's defenses are permanently, exhaustingly elevated. She has never been inside a boarding school. She does not need to have been. The policy found her anyway. The downstream consequences of chronic cortisol dysregulation are not subtle. They are measurable across multiple organ systems. Researchers at the University of Manitoba studying First Nations boarding school descendants documented what they call elevated allostatic load. Allostatic load is the cumulative biological burden of chronic stress, the wear and tear on the body's regulatory systems. In third-generation survivors studied in Manitoba, researchers found measurable differences in cardiovascular function, endocrine output, and immune system response. Compared to community controls with no boarding school lineage, the differences were statistically significant. This cross-border data, from a parallel Canadian system, corroborates the findings from U.S.-based Anishinaabe and Lakota cohort studies. The biological signal does not stop at the 49th parallel. So here is where the research stands. As of 2022, 3 living generations of descendants show measurable cortisol dysregulation traceable to boarding school attendance. The mechanism, FKBP5 hypermethylation, is documented, replicated, and peer-reviewed. The question is no longer whether epigenetic transmission occurred. The question is now, across how many people? And what does the data show about scale? The scale anchors are important here. There are approximately 7.4 million people of American Indian and Alaska Native ancestry in the United States. Researchers and the National Native American Boarding School Healing Coalition estimate that the majority of members of the 574 federally recognized tribes have at least one direct ancestor who attended a federal or federally funded boarding school. This is not an individual trauma story. This is a population-scale epigenetic event. Now, look at the epidemiological data and see whether the biology predicts what we find. American Indian and Alaska Native populations experience rates of post-traumatic stress disorder approximately 2 to 3 times higher than the general United States population. That statistic comes from the Indian Health Service and the National Institute of Mental Health. For years, researchers attempted to explain that disparity through contemporary stressors alone: poverty, discrimination, geographic isolation. But epigenetic researchers are now arguing that those explanations are insufficient. They are insufficient because they do not account for a stress response system that arrived already dysregulated, already recalibrated by a grandmother's experience in a dormitory 80 years ago. Historical trauma is not a background variable. It is a biological starting condition. This is where Dr. Maria Yellowhorse Braveheart's work becomes indispensable to understanding the science. Dr. Yellowhorse Braveheart is a Hunkpapa Lakota scholar at Columbia University's School of Social Work. She developed the historical trauma response framework for Lakota communities long before epigenetic research caught up with what she was describing. Her framework identified a cluster of responses in Lakota adults: unresolved grief, elevated PTSD symptoms, depressive episodes clustering around historical anniversaries. In 2016, her foundational framework gained renewed clinical attention as epigenetic researchers began designing studies specifically targeting boarding school descendant cohorts. What she had described in psychological and community terms, the sequencers were now finding at the molecular level. Her words carry a precision that the laboratory confirmed. She said it plainly: the historical trauma response is not a cultural construct, it is a physiological reality. Our bodies carry what our grandparents were not allowed to grieve. Not metaphor, measurement. And here is the dimension of this story that Western science is only beginning to reckon with. For Lakota, Anishinaabe, Cherokee, and many other nations, the idea that trauma passes through generations is not a new discovery. It is embedded in oral tradition and ceremonial knowledge that has existed for centuries. The boarding school system itself actively suppressed that knowledge, forbidding languages, ceremonies, and the very transmission frameworks that communities use to understand and interrupt cycles of harm. Epigenetic research does not replace that indigenous knowledge. What it provides is a language that Western institutions are legally and procedurally equipped to hear. When tribal elders said the harm was carried in the blood, they were not speaking poetically. The methylation sequencer in Ann Arbor confirms they were speaking with a precision that Western science spent decades developing the tools to verify. The 526 schools identified by NABS in 2025 operated across 37 states. The highest concentrations were in Oklahoma, Arizona, and New Mexico. Those states are home to the Cherokee Nation, the Navajo Nation, known to their own people as the Diné, and numerous Pueblo communities. These nations had the highest historical enrollment numbers, which means their descendant populations represent the largest available cohorts for epidemiological study. In 2025, epidemiologists overlaid school locations across those 37 states onto contemporary maps of American Indian and Alaska Native health disparities, cross-referencing Federal Indian Health Service data with the confirmed school registry. Diabetes rates, suicide rates, substance use disorder rates. Each school location functions as an epicenter, with measurable downstream biological impact radiating outward across 150 years. The correlation is not coincidental. The biology predicted exactly this distribution. Substance use disorder among American Indian and Alaska Native populations has long been analyzed as a response to contemporary conditions. But public health researchers now argue for a more grounded framing. When the brain's central stress command system is dysregulated, when your cortisol system is chronically elevated, substances that dampen the alarm become neurologically compelling. This is not weakness. This is biology seeking relief from a recalibrated stress system. And here is where the evidence becomes impossible to look away from. The poverty rate for American Indian and Alaska Native communities sits between 22 and 24%, one of the highest of any demographic group in the United States. Chronic poverty independently elevates cortisol. It independently promotes methylation of stress response genes. So researchers are now documenting a compounding effect. Descendants arrive with FKBP, 5-hypermethylation, inherited from grandparents' boarding school experience. They then live in chronic poverty environments that independently drive the same methylation mechanism. The inherited dysregulation is reinforced by the contemporary one. The thermostat, already reset by institutional policy, gets pushed further by structural economic exclusion. These are not separate crises. They are the same biological cascade, measured across different timescales. Now watch what happens when you map the school locations against poverty rates. The overlap is near complete. The communities with the highest historical boarding school enrollment show the highest contemporary poverty rates and the most severe cortisol dysregulation in descendant cohorts. This is not correlation hunting. This is the expected output of a documented biological mechanism operating over 5 to 6 biological generations. 150 years of operation, long enough for 5 to 6 full biological generations to have passed, each one potentially inheriting the methylation signature of the original institutional trauma. In 2023, the Indian Health Service formally acknowledged intergenerational trauma as a clinical diagnosis category. This was a landmark institutional recognition, updated into mental health service delivery guidelines for the 574 federally recognized tribes. The IHS cited epigenetic research as the biological evidence underpinning elevated rates of depression, PTSD, and substance use disorder in American, Indian, and Alaska Native populations. For the first time, a federal health agency was not treating these disparities as social problems to be managed. It was treating them as biological outcomes of a documented federal policy with measurable mechanisms. The institution that ran the schools is now reading the biology the schools produced. But there is a parallel stressor operating alongside the inherited cortisol dysregulation, one that researchers are only recently quantifying. As of 2024, approximately 90,000 sets of Native American ancestral remains were still held in United States museum and university collections. That is roughly the population of Santa Fe, New Mexico, held in storage rather than returned to the nations they belong to. For Lakota, Anishinaabe, Cherokee, Pueblo, and hundreds of other nations, the inability to conduct proper burial ceremonies for ancestors is not a cultural inconvenience. It is an ongoing acute stressor, a source of grief that does not resolve, that does not complete its cycle. Tribal public health researchers and epigenetic scientists are now documenting something precise about that grief. Unresolved ceremonial loss, the inability to properly bury and honor the dead, independently elevates cortisol in surviving community members. It compounds the methylation patterns already present in descendants of boarding school survivors. The schools took the living children. The museums took the dead ancestors. Both absences are measurably present in the same cortisol sample. In 2024, amendments to the Native American Graves Protection and Repatriation Act took effect. Those amendments accelerated the return of approximately 10,300 sets of ancestral remains from museum collections. Researchers and tribal health advocates are watching what happens next in descendant communities. The hypothesis, grounded in epigenetic theory, is that access to ceremony, cultural practice, and the completion of ancestral grief cycles may function as measurable biological interventions, not as comfort, not as cultural preference, as documented mechanisms for interrupting the cortisol dysregulation cycles the boarding school system initiated. This is where the science arrives at a finding with significant implications. If the damage is epigenetic, the question becomes, can epigenetic changes be reversed? The current state of the research is cautiously affirmative. Methylation tags are not permanent the way mutations are. Under conditions of sustained psychological safety, secure attachment, and reduced chronic stress, methylation patterns can shift over time. The thermostat can be recalibrated, but only if the conditions that originally reset it are systematically addressed. Language access, ceremonial practice, secure housing, adequate nutrition. These are not incidental cultural preferences. The epigenetic data frames them as biological necessities. Repatriation is not symbolism. Language revitalization is not nostalgia. They are, in the framework of this science, interventions with measurable, downstream, biological outcomes. The morning air in Ann Arbor smells like wet pavement and diesel from the research campus buses. Inside the laboratory, there is the faint chemical cold of refrigerated sample storage. A technician places the saliva sample into the methylation sequencer. The machine does not know the history it is reading. It simply reads what is there. FKBP, 5-hypermethylation, elevated baseline, blunted awakening response. The readout is clinical, the meaning is not. 80 years, 3 generations, 1 molecular tag. But the most revealing part of this story is still ahead in what the 2025 data tells us about scale. When NABS confirmed 526 schools in 2025, up from 408 confirmed in 2022, epidemiologists had to revise their population models. 118 additional schools means 118 additional epicenters of measurable downstream biological impact. Each additional school represents additional descendant cohorts who have not yet been studied, who may be carrying the same methylation signature without any clinical framework to explain what their bodies are experiencing. NABS has responded by calling for federally funded longitudinal epigenetic health studies tracking descendants across all implicated tribal nations. The 2025 report is not a historical accounting. It is a public health research agenda. Consider what a longitudinal study at this scale would actually require. 526 school locations, 37 states, 7.4 million people of American Indian and Alaska Native ancestry, descendant cohorts spanning 3 to 4 living generations for the earliest schools, saliva samples, blood cortisol panels, morning awakening response measurements, allostatic load assessments, cross-referenced against documented boarding school enrollment records for each of the 526 institutions. This would be one of the largest epigenetic studies in American medical history. It has not been funded yet. The data exists. The will has not arrived. The American Indian and Alaska Native PTSD rate, 2 to 3 times the national average, represents a calculable population health burden. Emergency mental health visits, hospitalizations, lost economic participation, shortened life expectancy. Public health economists are beginning to model what the boarding school system's epigenetic legacy costs annually in medical expenditures alone. The early figures are not yet published, but researchers say they are not small. The science also speaks directly to something that tribal communities have navigated without institutional support for generations. Children growing up in boarding school, descendant families often experience a parent or grandparent whose stress responses feel disproportionate, unpredictable, or impossible to soothe. A caregiver whose cortisol system is chronically dysregulated cannot consistently provide the regulated, responsive attachment that epigenetic research shows is essential for healthy cortisol calibration in the next generation. This is the transmission vector that researchers call attachment disruption. The boarding school system separated children from parents, interrupting exactly the relational framework that calibrates a child's stress biology. That interruption then repeated itself, generation after generation, as dysregulated parents raised children who became dysregulated parents. The institution designed the mechanism. The mechanism ran itself forward. In 2023, when the Indian Health Service updated its clinical guidelines, it formally named this cascade. Providers working with American Indian and Alaska Native communities were instructed to screen for intergenerational trauma as a biological baseline variable, not an anecdotal history. A clinician treating a Lakota adult for depression in 2023 is now trained to ask, does this patient's cortisol dysregulation have an epigenetic component traceable to boarding school ancestry? That question changes the treatment model entirely. Standard antidepressant protocols were not designed for a stress regulation system that was epigenetically recalibrated before the patient was born. The biology requires a different clinical response. Researchers studying therapeutic interventions in boarding school descendant communities are now testing whether culturally grounded healing practices produce measurable cortisol changes. The hypothesis is that language immersion, ceremonial participation, and intergenerational storytelling practices, the boarding school system explicitly and forcibly suppressed function as stress regulation interventions at the neurobiological level. The studies underway draw on community-based participatory research models, where tribal nations define the parameters, the populations sampled, and the outcomes measured. Preliminary data from those programs is showing cortisol normalization effects in participants engaged in sustained cultural practice. The sample sizes are small. The studies need replication and scale, but the directional signal is there, and it points towards something that tribal nations have known for generations. Healing the biology requires restoring what was taken from the biology's community of origin. The Anishinaabe woman whose saliva sample opened this story is not defined by what the sequencer found. She is a member of a community that has been naming this harm for generations, long before any laboratory had the instruments to confirm it. Tribal nations are not waiting for the federal funding to arrive. Language nests, healing circles, repatriation ceremonies, cultural immersion programs, built by communities, for communities. The biology of repair is already underway. The sequencer in Ann Arbor reads one tag at a time. The communities it draws from are already writing the next chapter. Federal policy created the methylation pattern. Living communities are the ones changing it. Language nests, healing circles, repatriation ceremonies, cultural immersion programs, built by communities, for communities. The biology of repair is already underway. The sequencer in Ann Arbor reads one tag at a time. The communities it draws from are writing the next chapter themselves. That question changes the treatment model entirely. Standard antidepressant protocols were not designed for an HPA axis that was epigenetically recalibrated before the patient was born. The biology requires a different clinical response. Researchers studying therapeutic interventions in boarding school descendant communities are now testing whether culturally grounded healing practices produce measurable cortisol changes. The hypothesis is that language immersion, ceremonial participation, and intergenerational storytelling practices the boarding school system explicitly and forcibly suppressed function as stress regulation interventions at the neurobiological level. Preliminary data from community-based studies is showing cortisol normalization effects in participants engaged in sustained cultural practice programs. The sample sizes are small. The studies need replication and scale. But the directional signal is there, and it points towards something that tribal nations have known for generations. Healing the biology requires restoring what was taken from the biology's community of origin. 526 schools means 526 distinct documented sites of these practices. 526 locations where the stress response systems of children were forcibly recalibrated. 526 points of origin for epigenetic cascades that are still running through living bodies today. 526 sites where the conditions for FKBP, 5-hypermethylation, were systematically created by federal policy. The buildings are gone or repurposed. The methylation tags remain. For the science audience watching this, it is worth pausing on what it means that